Hello friends, welcome to my YouTube channel Bio E-Content. Today we will explore RNA, an essential molecule of life. In earlier video we have discussed regarding the DNA, but today we are focusing on the next and very much important molecule, that is RNA. What do you think? What is RNA? How does it work? And why is it important in humans and plants? Let's find out. नमस्ते दोस्तों, हमारे चैनल में आपका स्वागत है। आज हम आरएनए के बारे में जानेंगे, जो जीवन के लिए एक महत्वपूर्ण अणु है। आरएनए है क्या? कैसे काम करता है? और यह क्यों जरूरी है? आइए जानते हैं। यू माइट बी थिंकिंग व्हाई आई प्रिपेयर्ड such video on RNA when so many videos are available on YouTube. But friends, I'm sure you will get the clarity from this video how the RNA works in humans as well as in plants. Before preparing the video, I fixed some objectives. First objective is I'm going to discuss the importance and role of RNA in humans and plants. Second objective of this video is the importance of the central dogma, जिसे हम केंद्रीय मत का महत्व भी कह सकते हैं. So let's introduce RNA. अभी आप लोगों को समझ में आ रहा होगा कि डीएनए एक डबल स्टैंडर्ड मॉलिक्यूल है और आरएनए एक सिंगल स्टैंडर्ड मॉलिक्यूल. So what is RNA? RNA stands for ribonucleic acid. It is a molecule that plays a vital role in genetics and protein synthesis. Unlike DNA, which is double stranded, RNA is usually single stranded. RNA का पूरा नाम राइबोन्यूक्लिक एसिड है. ये एक अणु है जो आनुवंशिकी और प्रोटीन संश्लेषण में महत्वपूर्ण भूमिका निभाता है. जैसे कि मैंने अभी कहा, डीएनए इज डबल स्टैंडर्ड यानी डबल स्टैंडर्ड होता है और आरएनए आमतौर पर सिंगल स्टैंडर्ड होता है. यू कैन सी हियर DNA is made up of two strands which are arranged or coiled in antiparallel manner, and the DNA is always made up of four bases. Those are A, T, G, C. Whereas the RNA is usually a single stranded, and it has nitrogenous bases like A, U, G, C. The one base difference is there in between DNA and RNA. Uric acid is present in RNA instead of thymine in DNA. Let's talk about the central dogma. आणविक जीव विज्ञान का केंद्रीय मत. Why I am explaining this before explaining the RNA? Because the central dogma is the central theme of any living organism where lacks of chemical synthesis or chemical reactions takes place. In chemical reactions, this part, that is central dogma, is very much important to make you healthy and to make you diseased or unhealthy. So let's discuss this one. Central dogma of molecular biology. What is it? How does it explain the flow of genetic information? Let's discuss. आज हम आणविक जीव विज्ञान के केंद्रीय मत के बारे में जानेंगे. यह क्या है? ये आनुवंशिक जानकारी के प्रवाह को कैसे समझाता है? आइए जानते हैं. व्हाट इज द सेंट्रल डोग्मा देन? केंद्रीय मत क्या है? The central dogma of molecular biology explains the flow of genetic information. You can see here, the genetic information is being sent from DNA to RNA. It states that genetic information flows from DNA to RNA and RNA to protein. This is the ultimate product. You can see the information will get transferred from DNA to RNA, then RNA to protein. आणविक जीव विज्ञान का केंद्रीय मत जैविक प्रणाली में आनुवंशिक जानकारी के प्रवाह को समझाता है. यह बताता है कि आनुवंशिक जानकारी डीएनए से आरएनए और फिर प्रोटीन तक जाती है. अब इस सेंट्रल डोग्मा में मेन कौन से स्टेप्स है? थ्री मेन स्टेप्स आर देयर इन सेंट्रल डोग्मा. Those steps we are going to discuss. First step is DNA replication. You can see here the DNA can make its own copies. यानी DNA अपनी आप प्रतिलिपि बनाता है. और सेकंड स्टेप है ट्रांसक्रिप्शन, जिसे हम लोग लिप्यंतरण भी कह सकते हैं. DNA is transcribed into RNA. DNA को RNA में परिवर्तित किया जाता है. और थर्ड है ट्रांसलेशन. RNA से प्रोटीन बनता है. That is RNA is translated into protein. RNA को प्रोटीन में अनुवादित किया जाता है. This is the theme of central dogma, and you must understand the mechanism of a central dogma. What is the importance of the central dogma? As I said, the central dogma is essential for life. If there is error in the central dogma, either it is in replication, in transcription, or in translation, you may suffer from diseases or disorders, etc. It ensures that genetic information is accurately transferred and expressed in cell. If the genetic information is incorrectly transferred or expressed in cells, you may have some abnormalities in your body. Both this is applicable in plants as well as in human beings. Any disruptions can lead to genetic disorders or disease. केंद्रीय मत जीवन के लिए आवश्यक है. यह सुनिश्चित करता है कि आनुवंशिक जानकारी कोशिकाओं में सटीक रूप से स्थानांतरित और व्यक्त की जाए. किसी भी गड़बड़ी से आनुवंशिक विकार या रोग हो सकते हैं. सेंट्रल डोग्मा, as we discussed, is the central theme, but in nature nothing is permanent. Some exceptions are also there. In central dogma also some exceptions are uh, available in the nature. How it is, we'll discuss. As we have seen, that DNA can make its own copies by using the DNA polymerase. This is the first thing. Then a DNA transcription, and from DNA to mRNA will get synthesized with the help of RNA polymerase. These are the enzymes required for uh, different stages. And if there is one enzyme, that is reverse transcriptase, is available, that from mRNA DNA may get synthesized. This is the exceptions to the central dogma. You can think about this. Some exceptions are also there in nature that we must discuss. The example is retroviruses in HIV. That is, retroviruses like HIV use the reverse transcription to convert the RNA to DNA. यद्यपि केंद्रीय मत एक मौलिक सिद्धांत है, इसमें कुछ अपवाद भी है. रेट्रोवायरस जैसे कि HIV रिवर्स ट्रांसक्रिप्शन का उपयोग करके RNA को DNA में परिवर्तित करते हैं. So friends, this is regarding the central dogma, which is playing a key role in molecular biology that explains how genetic information is used to produce the protein. Understanding this concept helps in genetics, biotechnology, and medicine. तो केंद्रीय मत आणविक जीव विज्ञान का एक प्रमुख सिद्धांत है जो यह समझाता है कि आनुवंशिक जानकारी का उपयोग प्रोटीन बनाने के लिए कैसे किया. इसे समझने से आनुवंशिक जैव प्रौद्योगिकी और चिकित्सा में मदद मिलती है. So friends, now we'll discuss the importance of RNA in humans and plants. How does RNA functions in our body as well as in plants? Let's find out. नमस्ते दोस्तों, आज हम मनुष्य व पौधों में आरएनए के महत्व और भूमिका के बारे में चर्चा करेंगे. आरएनए हमारे शरीर और पौधों में कैसे कार्य करता है, आइए जानते हैं. As we have discussed, RNA is a ribonucleic acid, is a crucial molecule that helps in the process of gene expression and protein synthesis. हम अब इस आरएनए के बारे में स्टेप बाय स्टेप डिस्कस करेंगे. What is the role of RNA in humans and plants? In humans and plants, RNA is involved in various essential functions. तो इस functions को carry out करने के लिए what are the different types of RNAs are involved? Those types we are going to discuss. पहला टाइप आरएनए का है mRNA, जिसे हम लोग मैसेंजर आरएनए कह सकते हैं. It carries the genetic information or genetic instructions from DNA to ribosome for protein synthesis. डीएनए से जेनेटिक निर्देश लेकर राइबोसोम तक पहुंचाता है और ताकि प्रोटीन संश्लेषण हो सके. सेकंड टाइप ऑफ आरएनए इज टीआरएनए, दैट इज ट्रांसफर आरएनए. It helps in assembling the amino acids to form, this is one, tRNA, to form the proteins. amino acids को इकट्ठा कर प्रोटीन निर्माण में मदद करता है. और थर्ड टाइप है आरएनए का आरआरएनए, जिसे हम लोग राइबोसोमल आरएनए भी कह सकते हैं. Forms the structure of ribosomes where proteins are made. राइबोसोम्स की संरचना बनाता है जहां प्रोटीन बनते हैं. अभी सबसे ज्यादा इंपॉर्टेंस जिस आरएनए को दिया जाता है मॉलिक्यूलर बायोलॉजी में, वो है एमआरएनए, एमआईआरएनए एंड एसआईआरएनए, माइक्रो आरएनए एंड स्मॉल इंटरफेयरिंग आरएनए. और ये दोनों को हम लोग रेगुलेटरी आरएनए भी बोल सकते हैं. इट इंक्लूड्स micro and siRNA, which regulate the gene expression and protect against harmful genetic mutations. यानी हमारे बॉडी में म्यूटेशंस ये प्रोसेस कंटिन्यूस चलते रहती है, लेकिन उस म्यूटेशंस को अवॉइड करने के लिए हमारे बॉडी में रेगुलेटरी आरएनए का होना जरूरी. मनुष्य और पौधों में आरएनए एक महत्वपूर्ण कार्यों में शामिल होता है. Just like what we discussed. You can see here the role of RNA, the messenger RNA. But I will again add something new in your dictionary. That is, the DNAs are present in nucleus. The DNA just gives the instructions as per the change in atmospheric condition or surrounding. Then that DNA gives the instructions to the for the formation of mRNA, and the actual protein synthesis will take place in the cytoplasm. Where
is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape.
In between these two molecules, DNA and RNA. DNA is usually having two strands, but in some rare cases, it may have one. In case of RNA, usually one and rarely two strands. The sugar molecule is present in DNA is deoxyribose, whereas in RNA it is ribose. Bases, as we discussed, are ATGC in DNA, whereas in RNA, AUGC. E is not there in RNA. Instead of thymine, uracil is present in RNA. Pairing is like that. A will always pair with the T and G will always pair with the G. In case of RNA, adenine will get paired with the uracil and G will be paired with the cytosine. The location of the important molecules, that is DNA, mostly it is present in chromosome, but some DNAs are present either in mitochondria and in chloroplast. But in case of chromosome, RNA, it is present in chromosomes and ribosomes. Replication, and how the copies of these molecules get prepared. The DNA is having the capacity to replicate itself. That's why the DNA is self-replicating. But in case of RNA, it must need it